Hello everyone, today we are sharing about the mechanisms of toxicity of naphthalene, that is, how naphthalene works in the body.
The main metabolites of naphthalene are 1-naphthol, trans-1,2-dihydrodiol, and 2-naphthol. Trans-1,2-dihydrodiol is produced in 10 times the amount of 1-naphthol, and 1-naphthol is produced in 10 times the amount of 2-naphthol. The reason for the formation of trans-2-dihydrodiol instead of the cis form of dihydrodiol is possibly due to the catalytic mechanism of the epoxide hydrolyse in humans. Glutathione, also abbreviated as GSH, comprises of 3 amino acids, cysteine, glutamine, and glycine. It binds to destructive molecules such as peroxides and free radicals. In the body, naphthalene causes GSH depletion by binding to it, reducing the available GSH to perform its other functions such as protein synthesis, prevention of oxidative stress on DNA synthesis, and metabolism of other toxins.
Lipid peroxidation is a process generated naturally in small amounts in the body, mainly by the effect of several reactive oxygen species such as hydroxyl radicals or hydrogen peroxide. These reactive oxygen species attack the polyunsaturated fatty acids of the fatty acid membrane, initiating a self-propagating chain reaction. The destruction of membrane lipids and the end products of such lipid peroxidation reactions are especially dangerous for the viability of cells, even tissues. Since lipid peroxidation is a self-propagating chain reaction, the initial oxidation of only a few lipid molecules can result in significant tissue damage. The effect of naphthalene on lipid peroxidation and antioxidant status was studied in the hepatopancreas, hemolymph, and ovary of the Xylus serrata, the giant mud crab, with reference to the active vitellogenic stage. Results showed that there was an overall increase in lipid peroxidation activity in the tested samples. Many types of cancer cells are also found to have increased levels of reactive oxygen species. For example, leukemia cells freshly isolated from blood samples from patients with chronic lymphocytic leukemia or hairy cell leukemia showed increased ROS production when compared with normal lymphocytes. An increase in ROS production will thus cause cancer in the long run, as ROS in high levels can easily interact with the lipids that make up the cell membrane and cause drastic changes to the selective permeability of the cell membrane. ROS can also cause damage and genomic instability to DNA and modify proteins such that enzymes may be catalytically less active than before.
So moving on to DNA damage caused by naphthalene. The naphthalene metabolites 1,2-NQH2, which stands for 1,2-hydroxynaphthalene, 1,4-NQH2, which stands for 1,4-hydroxynaphthalene, 1,2-NQ, which stands for 1,2-naphthylquinone, and 1,2-NQ, which is 1,4-naphthylquinone, all causing DNA damage. The metabolites of naphthalene 1,2 and 1,4-NQH2 cause damage in DNA when under presence of copper-2 ions. The treatment with piperidine significantly enhanced DNA cleavage, suggesting that DNA damage was due to base modifications alongside with strand breakage. In addition, adding NADH, which stands for nicotinamide adenine dinucleotide, increased DNA damage by 1,2-NQH2, but it did not affect the extent of damage done by 1,4-NQH2. So for oxidative damage to DNA by the naphthalene metabolites, 1,2 and 1,4-NQH2 are oxidized into their respective semiquinone radicals, followed by the oxidation into 1,2 and 1,4-NQ. So the first step of oxidation is enhanced by the copper-2 ion, where oxygen is reduced to O2-, which is then dismutated to H2O2, hydrogen peroxide, and reduced copper-2 ions into copper-1 ions. Copper-1 ions and H2O2 then induces oxidative DNA damage through the formation of CuOOH, which stands for cupric hydroperoxide. However, the conclusion that cupric hydroperoxide is formed is just merely a speculation. Cupric hydroperoxide appears to be responsible for the formation of piperidine label products and 8-oxo-7,8-dihydro-2-deoxyguanosine, which act as oxidative DNA markers. These results hence indicate that NQH2s are oxidized into semiquinone radicals, followed by their respective NQs, and that H2O2 and copper-1 ions are obtained during oxidation. 1,2-NQ is reduced by NADH to form the redox cycle, resulting in enhanced DNA damage. Therefore, this shows that the extent of damage done to DNA is larger by 1,2-NQ or 1,2-NQH2 compared by the extent of damage done to DNA by 1,4-NQ or 1,4-NQH2.
An experiment performed on C. elegans to show the possible inhibition of apoptosis by naphthalene as a non-genotoxic carcinogen proved mostly positive. Through counting extra surviving cells in the anterior pharynx of individual animals after the conduction of each round of the experiment, clear statistics will be able to be taken note of. Researchers grew some roundworms in an incubator with naphthalene mothballs and saw an average of 4 to 9 extra living cells in the anterior pharynx. This result suggests that naphthalene substantially enhances the cell death defect. Therefore, naphthalene does suppress cell apoptosis. In the mechanism of how apoptosis suppression is induced, it has been found that a naphthalene metabolite directly inactivates protease enzymes, caspases, that play a crucial role in apoptosis by oxidizing the active site cysteine, suggesting the molecular mechanism by which naphthalene suppresses apoptosis.